I'm Catherine, a PhD candidate in the Department of Evolution, Ecology, and Organismal Biology at the University of California, Riverside. And today I'll be presenting on the effects of age at first fatherhood on reproductive outcomes. So life history theory predicts that older animals should invest more in current reproduction than younger animals due to the decline in residual reproductive value with age. As a result, old animals may produce higher quality offspring than younger animals. However, as organisms age, they may also experience senescence, which is a decline in physiological function. And because of senescence, old animals actually might end up producing lower quality offspring than younger animals. One form of parental investment is parental care, and today we'll be focusing on biparental care, or care provided by both mother and father. Biparental care is beneficial in that it can reduce the workload on both individual parents, and in some species, the presence of both mother and father is required for offspring to survive. While biparental care is found across a wide range of taxa, it is only present in about 5 to 10% of mammalian species. As a result of the relative rarity of care by fathers of mammals, we know a lot about how age affects maternal care, but not so much about the effects of age on paternal care. I hypothesize that older fathers will be more paternally motivated than younger fathers, keeping in line with life history theory, and this will lead to higher quality offspring from old dads compared to young dads. To do this, I looked at the California mouse, which is a socially and genetically monogamous species of deer mouse. They are biparental, with fathers displaying all the same types of parental behavior as mothers, with the exception of nursing. They live in small family groups consisting of a mated pair and a litter of about 1 to 4 pups. And in the wild, these animals can live up to 18 months. And in the lab, they have survived to at least 3.5 years. All animals in the study were captive individuals housed in a laboratory setting for the entire duration. I had males of 2 different ages at the beginning of the study, old males that were 16 months old, and young males that were 4 months old. And these males were all paired to females that were 7 to 8 months of age. In total, I had 24 old male pairs and 25 young male pairs. A week before pairing, all males underwent a parental behavior test in which they were exposed to a 3 to 5 day old unfamiliar pup for 1 hour. A week later, they were paired with females, allowed to have a first litter, and at 11 days postpartum, all fathers were tested in an open field pup test. During the first 10 minutes, they were alone in the open field, and we observed the behavior that was in the field. In the second half of the test, we added a pup for an additional 10 minutes and observed the pup-directed and non-pup-directed behaviors. Once the father's pups reached 28 days of age, I measured body composition, so fat and lean mass of the pups using MRI, and the pups were weaned away from their parents. The parents were then allowed to have a second litter, which we observed for another 7 days. During this entire period, I took home cage video observations at 3 different time points each week. The pups were also weighed each week, and between the ages of 11 and 16 days postpartum, we checked to see if their eyes had opened. On the fathers, I collected blood on days 8, 11, 22, and 25 postpartum to measure basal testosterone levels. So, due to senescence, I predict that old fathers will take longer to produce a litter than young fathers. But keeping in line with life history theory, I predict that initially pups from old dads will be higher in quality or higher mass than young fathers. I also predict that old fathers will provide greater amounts of parental care than young fathers. And at weaning age, pups from old dads will remain higher in quality, so they'll have higher mass as well as higher body composition than those from young males. Looking at the mass of the fathers before they were paired, we did find that old males are significantly heavier than young males, which you can see from these pictures here. During the parental behavior test, we found that old males were faster to approach the pup than young males, and that young males spent longer durations of resting and sitting still without touching the pup than old males. However, there were no other significant differences in pup-directed or non-pup-directed behaviors, and this aligns with a previous study in our lab which we found very few differences between old and young sexually naive males and their responsiveness to pups. In terms of reproductive parameters, we found no significant differences from the time between pairing and parturition, litter size, litter sex ratio, and pup survival between old and young fathers. In terms of pup development, we'll first talk about pup body mass. And we see that when the pups were 14 and 21 days old, pups from old fathers were marginally heavier than pups from young fathers. And on day 28, pups from old fathers were significantly heavier than those from young fathers. However, looking at other measures such as fat and lean mass or the age at which the pups open their eyes, we didn't see any differences between pups of old dads and pups of young dads. In the open field pup test, in the first half, we see that old fathers traveled greater distances in the outer half, the center half, and in total compared to young males. And this would suggest that old fathers are more active in the open field, which is inversely related to anxiety-type behavior compared to young males. However, in the second half after the pup was introduced, we didn't see any difference between old fathers and young fathers in pup-directed and non-pup-directed behaviors. So going back to our predictions, we actually didn't see any difference in the time between pairing and birth or initial pup quality between old and young fathers. And currently we are analyzing the home cage videos to determine whether or not there is a difference in paternal care between old and young dads. We did, of course, see some differences in weaning mass between old and young fathers, with pups from old dads weighing more than pups from young dads. So overall, there are some differences in mass at weaning age, but so far very few differences in paternal behavior itself. There are some caveats. Firstly, this was done in a laboratory setting in which the animals received food and water at all times, there were no predators, and the environmental conditions were more or less constant. Therefore, our results likely differ from what would actually occur in the wild. Additionally, although our old males are considered old compared to their wild counterparts, they might not have actually reached senescence at the age of 16 months. As for our future directions for the study, as I mentioned earlier, we are still analyzing the behaviors in the home cage of not only the father, but also of the mothers to see if there's any difference in maternal behavior in response to an old versus a young mate. Additionally, we'll be beginning to start running testosterone assays on the blood we collected from the fathers. And with that, I'd like to thank all these folks here, as well as our funding sources. If you have any questions, please feel free to reach me at my email or my Twitter account. And thank you.